Good afternoon, everyone, and welcome to today's Food for Thought seminar. My name is Rachel, and I'm part of the Food for Thought team at FSA, and I'm also joined today by my colleague Paula. And before I introduce you to today's speakers, I'd like to remind you of just a few housekeeping rules. As this is a Microsoft Webinars event, you can't unmute yourself, and you can't turn on your camera, and the chat function is also disabled today, but we do, however, welcome any questions or comments you may have, and you can post these in the Q&A box, the icon for which should be visible on your screen. Any questions will be picked up by Paula at the end of the presentation. However, please feel free to ask questions using the Q&A box as we go along, and we can come to them at the end. We are always looking for recommendations of speakers to invite to future seminars. So if you have any suggestions, then please let us know. Our email address is foodforthought@food.gov.uk. And for today's seminar, I would like to provide a very warm welcome to our guests, Malcolm Burns and Gavin Nixon from the National Measurement Laboratory at LGC. Malcolm is head of the GMO Analytical Unit and Senior Scientific Advisor for Food Authenticity Testing. He has over 25 years of postdoctoral experience in the use of molecular biology approaches for food authenticity testing and is an internationally recognized expert on the identification and quantitation of genetically modified organisms in food products. He is also the manager for the UK National Reference Laboratory for GMOs in Food and Feed. And Gavin, a science leader in food analysis, is an experienced postdoctoral researcher with over 24 years industrial experience within the fields of molecular biology, method development and validation, measurement research and spectroscopy. So the last decade has witnessed significant advances in analytical technologies for food fraud detection. This evolution has been mirrored in terms of the capabilities and availability of portable point-of-contact technologies, but a lack of harmonization and guidance in this sector has precluded their universal adoption. So today's seminar will present recent results from an FSA-funded review of the potential for point-of-contact technology for food testing. It will provide an overview of key terms and technologies, trends and barriers, and have a set of recommendations to further develop a framework to support point-of-contact food testing. So with that, I'll hand it over to our wonderful speakers to discuss this research. And from all of us at the Food for Thought team, we hope you enjoy the presentation today.
Many thanks for that wonderful build up there, Rachel. And just bear with me, I will start to share my slides at this end. Okay, I hope everybody can see that. Again, many thanks, Rachel, for that introduction. As Rachel said, myself and Gavin are here today to give a talk entitled Recommendations for Developing a Framework for Point-of-Contact Food Testing. Again, many thanks to the FSA for extending the invitation to give this presentation, and also for members at Food for Thought and allowing us to support their series of webinars. A warm welcome to everybody this afternoon, for those of you online listening to us, and particularly so extending a warm welcome to other colleagues in the EU and our international participants as well. So we're going to give a talk on recommendations for developing a framework for point-of-contact food testing. This is based on a FSA project, FS900408, which was entitled Guidance for Point-of-Contact Technologies. And the individuals working on this project were myself, Susan Pang, and Gavin Nixon at the National Measurement Laboratory at LGC, where Gavin Nixon was the operations manager associated with the project, and I'm pleased to say he is here with us today. So in terms of the presentation this afternoon, I'll give a quick introduction to the project background, inclusive of the aim and objectives of the project, then go on to examine some of the key learning aspects associated with the project results, and then move on finally to the conclusions, including some of the recommendations and potential future work. So in terms of the project background, Rachel's has already provided a nice overview of this, but again, the last decade, I think it's safe to say, has witnessed a significant advance in analytical technologies for food fraud detection. So, for example, if we look at the nucleic acid field, this could include such things as digital PCR and next generation sequencing. Now, this evolution has also been mirrored, I think, in capabilities and the availability of point of contact technologies. So, whilst there is no universal definition of point of contact technologies for food analysis, these can generally be summed up as portable analytical instrumentation, which can be deployed at the point of sample testing throughout the food supply chain. And these often offer the potential to screen samples both quickly and cost effectively. Now, whilst the benefits of so-called point of contact instrumentation are relatively well understood, their successful application really has largely been confined so far to the clinical diagnostic and human forensic sectors. And for food analysis, a lack of harmonization and guidance in this area may provide a natural barrier for universal adoption of the POC instrumentation. So this project follows on from a previous DEFRA project, FA0178, which was entitled Assessment of Point of Contact Testing Technologies to Verify Food Authenticity. And this project, commissioned by the FSA on guidance for point of contact technologies, was commissioned really to further elaborate on the requirement to develop guidance to promote POC technology uptake for food analysis with a focus on use for official controls. So in terms of the project details, the project itself was commissioned in September 2023 and finished earlier this year in March 2024, so a 6-7 month project. And the operations manager, as I introduced earlier, was Gavin Nixon, who is a science leader at the National Measurement Laboratory here at LGC. And I'm pleased to say Gavin is online today. Mainly really wanted to introduce Gavin because he did all of the heavy lifting associated with the project itself, but also Gavin is available for the question and answer session this afternoon as well. So in terms of the project objectives, the aim of the project was to understand the potential for point of contact technology for food testing, for official controls, and also its application to the wider food sector. Objectives to encompass this aim really revolved around assessing the current state of the art and availability of POC instrumentation, including reviewing what technologies are involved, their current applications, where they're being applied to different commodities, examining the gaps and limitations, but also engaging with end users to see what the requirements of such technologies were. And the central aspects here really were engagement with key stakeholders for expert views to inform on the direction of travel of the project. Now, we're extremely grateful to a number of stakeholders which were consulted as part of this project. We've got a non-exhaustive list there, but this includes the competent authorities in the UK, including the FSA, DEFRA, and Food Standards Scotland, but also stakeholders involved at the UK accreditation, UCAS, MHRA, and PathSafe as well. From the clinical and pathology areas. Stakeholders also included international partners, inclusive of IAEA, the International Atomic Energy Agency, the government chemists as well, but also consultation with local authorities, port health authorities, trading standards, public analysts, and FAN centres of expertise, and our colleagues at FERA as well. So we took key learning opportunities from synergistic projects and initiatives. This included the previous DEFRA project, the Government Chemist Programme, the Food Authenticity Network as well. As I said, we engaged with international bodies as well, the International Atomic Energy Agency, but also to provide some notable shout outs here as well to UCAS, so the United Kingdom Accreditation Society, particularly so in terms of looking at such things as ISO 17025 accreditation for non-targeted food testing methods. Additionally, consultation with MHRA, which is the Medicines and Healthcare Products Regulatory Agency, which should produce published guidance on point of care testing devices in the healthcare and medicines area. And again, taking key learning points from those and seeing where they can be applied for the food testing. And also consultation with colleagues at PathSafe, which is the Pathogen Surveillance in Agriculture, Food and Environment project. This is a cross-governmental program, cross-governmental project, and looking at the work stream there on use of point of testing devices, diagnostic tools for detection of foodborne pathogens, and, for example, antimicrobial resistant microbes as well. So additional mechanisms for feedback included an updated literature review from 2019 onwards, a stakeholder focus group meeting involving a number of external participants from UK competent authorities, official laboratories, trading standards, etc. Also making use of online questionnaires, so reanalyzing and doing a deep dive into the previous project questionnaire from DEFRA, but also developing and publishing a new questionnaire comprising of 39 targeted questions, which we received 124 respondents in relation to. So the key aspect today then, the main aspect I wanted to talk about was really the project results. So apologies for the text-heavy slide here, but in terms of a definition of POC instrumentation, as touched upon earlier, there's no universal definition of point of contact testing for food analysis. The nearest equivalent appears to be point of care testing, or POCT, recognised in the health sector, which can be defined as medical testing at or near the site of patient care by specifically trained, for example, healthcare or non-laboratory professionals. Now, whilst there's no universal definition for food analysis, POC term is generally understood to include portable analytical instrumentation, which can be deployed at the point of sample testing throughout the food supply chain, often offering the potential to screen samples both quickly and cost effectively. Now, again, I think it's safe to say there's a plethora of technologies and instrumentation which can facilitate such point of contact testing. The number of these is continually increasing, and they continue to increase as well in terms of advances which continue to be made in terms of miniaturization and portability of analytical equipment. Now, this diversity is also mirrored in terms of where the devices are being applied, not just for food authenticity testing, but also for quality and safety aspects. And additionally, that's also mirrored in terms of the range of commodities which POC devices are used to test. So again, for example, ranging from meat authenticity testing to, for example, allergen detection in herbs and spices mixes. So the plethora of these options for POC testing in the food sector is so diverse, really, that this often precludes universal adoption of a single set of guidance. Now, there's certain aspects of infrastructure that can be applied across the range of point of contact testing devices, but again, the diversity of the devices and where they're being applied often means that it's difficult to have a single set of guidance applied to each and every one there. So for key technologies, instruments and trends, I'm going to report on some summary findings here. Again, these are just general trends which are reported here. For any detailed information on this, we'll direct listeners today to the report, the publication on this, which I'll refer to at the end of the presentation. But generally, there's 5 sort of general groupings for this sort of key technologies. This includes rotational vibrational spectroscopy platforms, inclusive of near-infrared, Fourier transform infrared and Raman. Also spectral imaging platforms, inclusive of multi and hyperspectral imaging, mass spectrometry, nuclear magnetic resonance, and finally biological analyte-based platforms, for example, really being protein and nucleic acid-based ones, examples being ELISA and PCR. Now, the results from the literature review in terms of the key technologies pointed towards the most common technology being used being near-infrared spectroscopy. Again, a number of advantages associated with that, for example, being non-destructive. And that appeared to have broad applications across the authenticity, safety and quality areas associated with food analysis, but also broad application across a wide range of commodities. Recent years have also seen some trends there as well. Raman comes up quite a lot as well, where there's been a number of studies and reports on developmental devices and their applications there. I think portable and miniaturized multi and hyperspectral imaging instruments have also seen increasing traction over the last few years. Protein lateral flow tests are still being used, not just, for example, for detection of allergens or pathogens, but also there's some evidence there of them being used for meat speciation. And DNA approaches, whilst being more complex, do tend to have a range of applications, and more portable devices are being developed. The questionnaire responses. Now, this was a questionnaire where we had 124 respondents across a wide range of sectors, including food safety managers, official controls, retail manufacturers, etc. The results from this for trends in key technologies also mirrored that from the literature review. Around 1/3 of all respondents said that they used POC testing within their organization. This was most often applied upon receipt of a sample, for example, at a factory or a warehouse. Fewer respondents said that they used it at source, and there wasn't really much evidence of it being used currently at port of entries. But currently employed POC technologies appear again to be mainly represented by spectroscopic techniques, for example, NIR again, and also immunological approaches, for example, ELISA. In terms of trends on the applications, some key areas that came to the front really were where the POC technologies were being used in, for example, repetitive testing applications. So for examples of large shipments where you have, for example, a large shipment of grain or rice which needs to be repeatedly tested at different parts of the shipment. Additionally, there was evidence of POC application and its utility as a portable and real-time instrument for sampling hotspots. And again, what do we mean by this? Well, again, looking back at the sort of shipment container aspect of for example, some grains, or whereby we might have a heterogeneous sample, whereby we may have some sort of layering during the transit. We may have different sort of sediment at the top and the bottom, and again using POC instrumentation to focus on these sampling hotspots. Another aspect in terms of applications was the non-destructive POC approaches, which have got clear utility for screening high value commodities, particularly, for example, herbs and spices. Again, an example might be saffron, where on a weight per weight basis, saffron is actually more expensive than gold and also subject to adulteration. And finally, something also mentioned by our official control stakeholders was the use of POC technologies really as cost-effective screening approaches, looking at those in terms of a balance between what they can achieve, but also awareness of potential limitations on the specifications or the scope of having a miniaturized piece of equipment. In terms of trends for testing on commodities, there were no real surprises here. Again, topical commodity and food authenticity testing seemed to remain consistent with previous years, whereby the top-rated commodities included using POC technologies to look at meat and fish speciation, adulteration of herbs and spices, and testing for allergens. Recent years, I think, has been a trend which has been further augmented, not just for looking at authenticity testing, but also quality and safety applications. So there's some evidence there of POC technologies being used for looking at moisture content, firmness, and even in some instances, chemical composition. Also looking for industrial chemical residue testing, for example, pesticides and insecticides, and some evidence for using them for looking for such things as food spoilage, aflatoxin, and presence of antibiotics. There was also some evidence of POC technologies being applied for looking for geographical origin or protected designated origin testing. There were some cited examples of ones being used for geographical origin for honey, coffee beans, cheeses, spices, alcohol, etc. Although I would probably say that these are more in the developmental stages as opposed to confirmatory testing. So, advantages and benefits associated with point of contact testing for food analysis. These are generally well understood. They revolve around a number of aspects. For example, the portability, the size and weight of the instruments themselves. They've got the capability to provide rapid and real-time results, often used as screening applications. Some of the technologies and instruments offer a non-destructive approach, so you get the sample back afterwards as well. Some of them are based on a non-targeted and multi-analyte approach as well, and a lot of them often offer cost savings, for example, being used as screening devices. An additional role, which was commented on by official control stakeholders, was that the real-time capability of some of these POC devices offer the potential to provide an effective tool for monitoring the food supply chain, which could, for example, facilitate timely intervention to stop any product distribution, sale, or consumption where it's necessary, for example, enabling timely quarantine and removal of any suspect products from the market. In terms of barriers and limitations to the uptake of POC technologies and instrumentation, any analytical approach, be that facilitated through a lab-based instrument or even the POC devices here, the efficacy of these approaches is completely dependent upon the availability of appropriate reference materials and open access curated databases. I think it's also safe to say that the true portability of some instrumentation is yet to be fully realized. A lot of devices can be handheld and easily transported, whereas some claim to be portable but may require quite a bit of heavy lifting, or may, for example, fit in the back of your car but not in the palm of your hand. Other barriers and limitations include comparability results. How do we compare results between instruments, between technologies? One of the limitations appear to be any lack of validated methods, but not just a lack of validated methods themselves, but the lack of validation data to support some of the instrumentation. Another barrier was, for example, multiplicity of testing. This is the idea whereby purchasing just one instrument where it can be used across different applications and different commodities would be of benefit. Costs for POC devices is also a factor as well, and not just the purchase of the initial instrument itself, but also its maintenance, its upkeep, and also such things as consumables, reagents, and the need to acquire and maintain accreditation. Some of the stakeholders obviously saw the application of POC devices just as screening approaches, and equally well just qualitative capabilities, so presence or absence, as opposed to full quantitation and compliance with, for example, a relevant threshold value. Support mechanisms which can be put in place to help these and overcome the barriers and limitations include, for example, production of appropriate reference materials, so that this would enable results to be effectively compared relative to a common reference standard. Again, development of databases is important, but equally well to have these as open access curated databases to provide a level playing field for stakeholders when we're trying to compare results. Training is also important, not just on the application of the instrument itself, but also how to interpret results. And that was a point further emphasized by official controls. It's important to be able to harmonize and compare results as well. And as touched upon, it's not just funding for acquisition of the instrument itself, but training and maintenance of the methods, which was also commented on by official controls. One interesting aspect was what's the feasibility of, for example, providing a centralized function based a sort of function based on looking at POC testing and providing an advisory framework from one central body there. And also support mechanisms include focus on technologies and instruments which facilitate multi-analyte capabilities, so looking at different targets as well, but also looking at multiplicity of testing. So, for example, one instrument which could be used for testing for multiple authenticity, safety, and quality applications as well. Another aspect which was commented on was that the area needs to be intelligence and trade flow led rather than instrument and technology led. So what do we mean by this? And again, this is something which was commented on by our official control stakeholder community. The idea here is that we look at what the actual issue is, for example, in the food supply chain, what are the emerging aspects there, what actually needs analysing, and then we fit the instrument to the issue at hand, rather than, for example, having the instrument developed and then saying, where can this be applied? So it really needs to be intelligence-led rather than instrument or technology-led. Of course, a further aspect of support mechanisms were mentioned were method validation. And again, that's important to provide objective evidence that a method is fit for its intended purpose. And this can be, for example, as part of ISO 9001 certification and compliance with a approved quality management system, ISO 17025 accreditation, so method specific, but also complying with international guidance and standards in this, inclusive of the IUPAC guidance and Eurochem as well. This was also commented on by official controls. The importance of having single laboratory validation, followed by interlaboratory collaborative trial where necessary. So not only to characterize the repeatability of the instrument and the method, but also its reproducibility as part of a collaborative trial. So assessment of important performance characteristics. This would include, for example, your traditional performance characteristics of trueness, precision, selectivity, sensitivity, but also, for example, particularly in relation to POC technologies, elaborating further upon the scope and error rates. What are the false positive and false negative error rates there? Also important to look at is the establishment of guidance on critical steps and minimum performance criteria to show that a instrument is fit for purpose. And finally, also being aware of how the results are produced and characterizing the measurement uncertainty associated with those results. Now, whilst we've talked about classical method validation aspects, another additional aspect, particularly associated with point of contact technologies, was ConOps, or the concept of operations. Now, again, this was important in terms of not just classical-based validation parameters, but also looking at end user requirements. Now, we've touched upon this earlier, but this also includes, for example, taking into account the expense of the instrument, not just the initial instrument cost, but its maintenance and servicing, reagents, consumables, etc. What is the availability of instruments as well? What is the availability of training and expertise to help out if there is an issue with using the instrument? How easy it is to use the instrument? What is its size, weight, and true portability? Do they offer rapid real-time results? What is the time to provide an analytical result? Most of the devices are qualitative, but some may offer some quantitative potential, so compliance, for example, against a relevant legislative threshold. What food types and commodities can the instruments be applied to? What is the complexity of sample preparation and representativeness of the sample being analysed, particularly so if you're using a miniaturized device? And also another aspect of ConOps, which was mentioned, is the results format and interpretation. Can this be harmonized and standardized, and how easy it is to interpret and look at the results themselves? Some final aspects were also commented by official controls included the requirement for a cost-effective simple test. So again, looking at the cost of the POC devices and maintaining those, but also commenting that affordable POC instrumentation could, for example, facilitate triaging and allow focus on more urgent areas for official controls. So, for example, could screening POC devices used to screen some targets of interest just to see if there is an issue or not associated with the production of a sample. If there isn't, that's fine, continue production, continue importing. If there is, could we then send that off for confirmational approach in a laboratory setting? Linked with that is the potential for POC devices to mitigate costly lab-based analyses. If we're using a portable device to actually check the integrity or compliance of a sample, that may actually free up more time in the laboratory-based setting to use some of the more costly devices for other urgent areas. And accreditation was also seen as a central aspect for future uptake, not just the cost of acquiring accreditation, but also maintaining accreditation for the future. So, conclusions and the road ahead then. So, a summary of the key recommendations. So, as we said earlier, the diversity of options for POC testing in the food sector is so large that this often precludes universal adoption of a single set of guidance or method validation requirements. So, one of the recommendations is that validation guidance needs to be provided on a specific POC technology, instrument, application, and commodity combination. Again, method validation is instrumental and fundamental in looking at the fitness for purpose of a method, but the method validation should not just be confined to the laboratory-based environment, but also should also take into account field-based setting at the point of application. As with any analytical approach, the development of appropriate reference materials and open access curated databases was seen as fundamental in helping underpin the infrastructure for this and facilitating and enabling the proper comparison results relative to a common reference. And as well as the more traditional method validation approaches, the evaluation of the end user requirements, ConOps, was very important. And we touched upon this earlier, but again, a centralized UK-based POC testing and advisory framework was mentioned by a number of stakeholders. That should be considered, which could, for example, offer harmonized training, independent advice, assessment of cost effectiveness of POC methods, and advice on how to maintain and curate open access databases. And overall, all of these key recommendations strive towards helping evaluate and characterize POC instrumentation for food testing, which itself supports the central theme of the FSA strategy 2022-2027 in terms of the central theme of food you can trust, helping enable consumer choice. So suggested further work includes, for example, whilst POC testing was a very broad area, looking at validation and guidance, which may have to be technology and instrument specific. So for example, using a case example, almost in terms of a business case, based on some of the more promising POC instrumentation and commodity combinations, that could be used as a candidate for method validation as an illustrative example, clearly trying to demonstrate the utility of that instrument and technology combination, as well as potential cost saving implications there as well. And again, if that were to be taken forward, the literature review, stakeholder engagement, the questionnaire, etc., pointed towards suggesting that NIR-based instrumentation would be a good candidate for that, for example, coupled with topical commodity testing, such as meat, fish, or herbs, spices, or allergens. And again, a comment very validly made by our colleagues in official controls was that further engagement with regional groups in the UK associated with official controls should be sought in order to canvass any additional views and regional variations for POC requirements. So finally, here's an example of the report, the publication front page, and there's a link there where you can access the final report, which provides further detailed guidance and breakdown of the review and recommendations we did. And if anybody would like to find out a little bit more, please feel free to access the Food Authenticity Network. If you go through to the main page and then click on the e-seminars as part of the training section, there is an e-seminar on a related topic there in terms of point of contact devices for food safety, authenticity, and quality testing. So it turns to me now just to provide a few acknowledgements, just to acknowledge Susan Pang at the National Measurement Laboratory in LGC for her contributions. and efforts towards making this work a success. Also Sophie Brooke, who was at the FSA as the project officer, but also to Rachel Pointer and Paula Monahan-Key today, as well as part of the FSA for hosting this webinar. And we specifically like to thank a number of other individuals who were involved as stakeholders associated with the project itself. So many thanks to everybody on their screen there. And also many thanks for all of your time. Both myself and Gavin are now available to take any question and answers. So many thanks for everybody's time.
Thanks so much, Malcolm and Gavin, for a fantastic and really interesting presentation. We will take some questions now, so if you do have any for our speakers, please make sure that you pop them into the question and answer box. But we do have some questions already in there, so I'll just go through some of them with you. So the first one we have is, what is defined as portable? Would a benchtop size NIR Spectro be considered portable, as often stated by manufacturers?
Yeah, I'll pass this over to Gavin in a few seconds, but yes, it's difficult to tell really. I think the portability would mean that you can easily take it along to the point of testing in the food supply chain, as opposed to, for example, it being a weighty benchtop size instrument whereby you have to take the sample along to the actual sort of laboratory facility itself. Again, I don't think there's a universal understanding or adoption of the word portable for this. I think for point of contact technologies, we're mainly talking about things which can be easily handheld, are light, and can easily be taken along, as opposed to something which claims to be portable, but for example, can only be lifted by 3 or 4 strong people and put into the boot of your car. Gavin, if I could perhaps pass over to you based on your knowledge of the sort of technical area as well, if you've got any further comments on that.
Yeah, of course. Thank you, Malcolm. So yeah, with regards to portability, yes, there can be a little bit of wriggle room, I suppose. Often it can be classified as transportable, as in rather than you can easily hold it in your palm of your hand, as Malcolm was saying, you could actually have it in a box and potentially kind of take it to the point of test. It's also dependent on the kind of factors such as what kind of sampling and kind of potential extraction resources are required around the actual instrumentation. For classical point of contact, you would normally expect it to be nice and simple, something to be very simple, kind of point and test. That's essentially the approach you want. If it's very complex and requires a lab around it, I wouldn't consider that kind of point of contact portable device as such. So it's kind of application driven, but yes, portability is the way to go. And if it's a very large complex instrument that requires a lot of supporting instrumentation, a lot of reagents around it, I probably wouldn't consider that as truly portable. So.
Perfect, thank you very much. So we have another question here. Dairy products are generally measured for protein content, which is represented by nitrogen content. If some companies use melamine to add into the measurement of nitrogen element will be very high. Your technology can detect the added chemicals in it.
Okay, so that's slightly beyond our sort of field of expertise. Myself and Gavin are molecular biologists by trade, looking at nucleic acid, and the point of the exercise really was to assess the applicability of a wide range of POC instrumentation there. It's certainly a valid question that we can get back to sort of offline as well. We have a number of expert colleagues at LGC, and as part of the government chemist function, that we can help provide an answer to that. Yeah, it's beyond my current knowledge. Gavin, I don't know if you'd be prepared to provide any comment on that at all.
So there are kind of systems out there that may be suitable for kind of melamine detection within kind of matrices such as dairy products. The kind of classical ones are kind of NIR based, kind of FTIR. So there are actually devices out there that can be used for melamine. Obviously, there are kind of chemical approaches as well. It's just kind of making sure that it's kind of appropriate for the kind of matrix and making sure it's appropriate for the measurement that you want to undertake as well. So yes, there are options out there on the kind of potentially portable side, but they do need a lot more kind of development and validation associated with them.
Yeah, and
Okay.
just to come back on that as well, this is clear evidence of why we have Dr. Gavin Nixon on the call today, as his technical knowledge far surpasses mine. But Gavin did pick up on a touch on a really relevant point there, which is validation. So irrespective of what instrument you're using, be that benchtop, be that point of contact, etc., the instrument needs to be validated. So we're really looking at, for example, the process of generating objective evidence to support the fitness for purpose of a method through the assessment of key performance characteristics. So there's a number of for example, standards and guidance out there which should be abided by. Again, I touched upon earlier the point of a quality management system. So ISO 9001 certification and an ISO 17025 accreditation, so that's method specific accreditation. That really needs to be looked at, but by going through the process of method validation, looking at important performance characteristics such as trueness, precision, specificity, sensitivity, and particularly I think for POC instrumentation, looking at the false positive and false negative rates, because potentially with a miniaturized set of equipment, you may be compromising on the specifications. So it's important to fully characterize the limit of detection and the false positive and false negative rates there. Through provision of that objective evidence, that should therefore allow you to see if an instrument is fit for purpose and, for example, what its scope of application is as well. And that's sometimes lacking when some of the POC instrumentation is available. Obviously, it's up to the laboratory to validate the method, but it's also important, for example, with instrument manufacturers as well, if we can access some of their validation data as well. Thank you.
Okay, so we have another one here. So they said, very interesting, thank you. There's a lot of discussion about the instruments themselves, but did you get a feel for how many of these tests were simple commercial lateral flow assays? And in brackets, that could be easily used by industry, for example, and how the fitness for purpose of these simpler approaches were generally perceived?
Sure. Again, I'll invite Gavin to provide a comment in a few seconds regarding that, because we may have had a little bit sort of a more informed response via the questionnaire as well that was part of the project. But certainly, I think some of the simpler tests were mentioned and continue to have traction in the area, whilst, for example, such things as NIR seem to be having increasing favour at the minute, mainly, for example, because it's non-destructive as well. It has applications across sort of authenticity, safety and quality aspects, as well as across different commodities as well. I think some of the lateral flow tests are still very useful. They're simple to use, simple to apply, and very cost effective as well. Gavin, do you have any sort of further comment on some of the simple POC devices or applications?
Yeah, there's kind of classical lateral flow devices. They're easily accessible, easily understood, easy to use, which is great for kind of POC applications. They've definitely got a kind of strong presence within the kind of testing space as well, and they've got a lot of kind of historical kind of context as well. So they're well understood. People understand how best to apply them and the associated kind of measurement performance. So the sensitivity, specificity, such, and how well it can be applied to certain matrices. So yes, they've got a very kind of strong role within the POC, the kind of in testing environment. That's not to exclude other ones such as kind of NAR based, which are still kind of for the food testing area are still being kind of developed to some extent, whereas the kind of lateral flow are kind of well established in the testing community.
Yeah, and again, to come back on that as well, to supplement what Gavin was saying, again, everything revolves around sort of method validation, showing that something is fit for purpose. But additionally, I think during the presentation itself, we touched upon the idea of the sort of ConOps, the concept of operations. So again, a lot of the stakeholder feedback said, yeah, you can do your classical validation approach, but it's important to recognize what is the ease of use of the actual POC device or instrument itself, as Gavin was saying. So what is its size and weight? What is the time to result? How available is it? How good is it at sampling? What is the results format and interpretation? So there is a lot to say about using some of these simpler devices, which are, for example, potentially less error prone, but also easier to apply and interpret. Thank you.
Okay, so we have which point of contact instruments are being used widely or successfully for food fraud purposes, and what products and by who is known. I'm thinking country of origin and speciation for products of animal origin and misrepresented herbs, spices, honey, etc.
Okay, so again, I think we'd probably refer people to the full report in relation to this, which has got a lot more detail in terms of the actual sort of instruments, the technologies, the breakdown of where they're being applied, etc. Again, such things as the NIR approach was being used for a number of applications and commodities there as well. I think for country of origin labeling, even with even with the benchtop based approaches, so the larger instrumentation, which have got a lot of better sort of specification, etc. I think country of origin labeling is still achievable, but it is not as well developed, for example, as looking at adulteration aspects associated with foods as well. So I think the state of the art is that it's still a question as to what is the best approach for country of origin labeling or protected designated origin. And I would suggest, for example, a weight of evidence approach using different across different sectors. So looking at, for example, a genomic approach, a proteomic approach, any imaging as well, building up the bigger picture to provide a weight of evidence approach may be the best one for country of origin labeling. Gavin, I don't know if you'd like to come in on that, if you've had any experience based on any aspects from the questionnaire or stakeholders in relation to that.
In relation to PBOs, I suppose the main focus of it was kind of classical food authenticity testing, so adulteration and such. There are kind of applications, I think, in development, as Malcolm kind of discussed, in the kind of herbs and spices, the kind of honey, kind of high value, kind of easy to adulterate, but suitable for kind of screening approaches. That the good thing about POC-based devices is that they're well is the good applicability to kind of screening, but then you need to also kind of consider what kind of downstream kind of confirmatory based approaches you'd need as well. So having an kind of total integrated system is probably the best way to see it, rather than just complete reliance on, let's say, POC. You would need to understand how it kind of feeds into your downstream testing as well for confirmation. So.
Yeah. And again, validation is the key aspect there, qualifying what the scope of application of a particular instrument or method is. And as Gavin was saying, I think, a lot of the stakeholder feedback recognize the benefits and potential use of POC instrumentation, but a lot also said, well, we recognize that miniaturizing something, you're potentially compromising on the specifications there. So a lot of times it could be used as a cost-effective and simple screening exercise. Is there an issue there? Yes or no? No, there isn't. Continue production, etc. Or yes, there is. Can we then send that sample onto a lab-based test or a benchtop-based test for the confirmatory test there as well? So it's really recognizing the balance between what the POC instrument can actually achieve in a field-based setting compared to, for example, what are the false positive, false negative rates, and is there a need for the additional confirmatory testing associated with that as well? Thank you.
Okay, so I think we'll just go for one more question because we're getting close to time. So someone has just asked, is there any views on future work needed for point of contact guidance?
Yep, that's a really good point as well. I'm happy to sort of field that one as well. In terms of the future aspects, then really, I think we sort of roughly touched upon those as part of the actual sort of presentation itself. I think again, perhaps an illustrative example would be useful, using a candidate POC instrument coupled with a particular commodity or sort of tria or application as well, really just to qualify the utility of a POC instrument in terms of validating it, looking at performance characteristics, and also qualifying the potential cost saving implications there as well. I mean, we've talked a lot about the sort of theory behind it, but actually provision of objective evidence to show that a POC instrument is fit for purpose is very important. And I think again, a lot of the feedback we got was that, well, it's a huge envelope POC testing. You're looking at multiple instruments, multiple technologies, different applications, different commodities. So one set of guidance does not fit all, but is there a way, for example, of having a centralized set of advice and guidance there in terms of, could we develop, for example, a framework providing a unified and harmonized set of responses and advice and databases and reference materials in relation to that? Gavin, is there any other aspects that you'd like to sort of comment on in terms of potential sort of further work? What else needs to be done in this sector?
Yeah, kind of following on from your comments, Malcolm. So yeah, kind of critical to kind of uptake and kind of further developments are kind of dependencies critical to a lot of the POC devices, especially within the spectroscopics field, where you've got kind of libraries, kind of spectral libraries, you've got databases, making sure they're validated, making sure they're curated, making sure they're representative of the samples being tested, and disseminating that, kind of disseminating information to the broader community as well, so that it's not compartmentalized within kind of organizations as such, making it available to the UK as a whole. Also kind of things such as kind of training, that's obviously very important as well, to actually try and kind of enhance overall kind of understanding and usage within the UK, and also kind of areas such as Malcolm said, the kind of validation, kind of helping to establish the kind of validation framework so that people can effectively use kind of POC devices as part of their kind of routine testing, and kind of demonstrating its kind of applicability in a field setting, because it's all well and good looking in that kind of lab setting where everything's controlled, but actually using it in the field, the kind of point of use, that's kind of critical to understanding and helping to support the uptake of these technologies.
Yeah, and just to sort of finish that off as well, if I may, Paula, just for the final sort of couple of minutes, having had time to reflect on that as well, obviously we had very good and constructive feedback from official controls as well in relation to the project, and the project was aimed at the wider testing analytical community, but also at official controls. And I think, you know, we need to acknowledge one size does not fit all for an effective solution. And whilst we're obviously trying to be representative during the project, it was just a small project. It really was just a sort of snapshot of some of the POC requirements as informed by expert official controls. And I think some of the feedback was, you know, we need to be aware of further engagement, for example, with regional groups in the UK associated with official controls, in order to, for example, canvass additional opinions and views and regional variations for POC instrumentation. So again, that's acknowledging that, for example, different areas, different local authorities may have different sort of requirements for sampling and analysis based on their years of experience and expertise of operating in that field. So again, for example, any regional variations in uptake or deployment of bespoke POC instruments, for example, in ports compared to large urban conurbations as well. And that would help us make a more informed decision on representative needs and requirements for POC instrumentation for official controls. We also briefly touched upon quantitative capability. Again, quantitative capability is important, for example, for looking at compliance with a legislative or a legal limit, for example, when looking at such things as pasta, basmati rice, GMOs, or a level for further enforcement action with meat or fish speciation. Again, from that technical angle, I think that needs to be further explored to help with POC instrumentation uptake. And as Gavin said, a sort of centralized UK-based POC testing and advisory framework would be useful, really to sort of harmonize how advice is being produced, looking at validated methods, disseminating those ideas as well, and that framework could play a sort of key role in helping maintain appropriate reference materials and curated databases, and thus helping ensure comparability results within the testing community. Thank you.
Perfect. Thanks so much. I appreciate there was a few questions that we didn't get to ask just in the time that we have. Malcolm and Gavin, would you be willing for people to contact you if they would like to ask those questions directly, because you had given your email addresses?
Absolutely, Paula, that should be fine. Alternatively, happy to take the sort of, Q&As as the sort of, text that came through during the actual sort of presentation as well, and we can respond to those offline as well.
Perfect. So I just want to say thank you so much for all the questions, and again to yourself, Malcolm and Gavin, for giving your time to come and present to us today. But before we close, I'd just like to say that Rachel has put the link into the actual full report into the question and answer box there for anybody who would like to see that. If you're not already on the Food for Thought mailing list and would like to join the mailing list, you can do so via the link that will go up into the question and answer box now. And recordings from some of our previous seminars will also be available to watch back on our YouTube channel. And again, that link will go up into the chat. And we would ask, we welcome ideas for future speakers. And if you are at the FSA and would like to get involved in volunteering in the Food for Thought team, please do get in touch. If you would like to contact us, please email us at foodforthought@food.gov.uk, or if you're at the FSA, you can search for our page on the intranet. But that just leads me to say thanks again. Malcolm and Gavin, thanks for joining, and enjoy the rest of your day.
Thank you.
Okay, thank you. Thanks, everybody.
Right, bye.